Dear students, I am Chandana Sengupta. Welcome to BioRatna. We are discussing the first chapter of genetics, that is Principles of Inheritance and Variation. We are now at the topic Chromosomal Theory of Inheritance. What is chromosomal theory of inheritance? You know that Mendel, he published his work in 1865. You also know that his work went unnoticed and he died without seeing that his work was recognized. In 1900, three scientists, they independently discovered Mendel's law of inheritance, that is Correns, De Vries and von Schirmack. First, we have to see why Mendel's work went unnoticed. It was a great work, undoubtedly. We call Mendel the father of genetics. Then why his work went unnoticed? There were many reasons for that. First one was that at that time, the communication was not easy. The communication was not that fast as now. This is one thing. Second, Mendel's factors, what Mendel said, that factors are the discrete units. Factors are the discrete units which control the characters. At the same time, Mendel said that each factor has two alleles. So, factors are the discrete units which controls the characters and a pair of alleles which do not blend. A pair of alleles which do not blend. There are many points here, na? Remember all. Do not blend. This was not accepted. This was not accepted as the reason for not accepted. Not accepted as the reason for continuous variation. Continuous variation present or observed in nature. Nature में जो ये continuous variation observe करते हैं हम, उसका reason they didn't, the scientists, the contemporary scientists, they didn't accept that it is the factors or this pair of alleles. Third, Mendel's application of mathematics, his application of maths and statistics in biological experiments was totally new, and this was also not accepted by the scientists. Now fourth, this is related to chromosomal theory of inheritance. This point, now listen carefully, then we will relate it to the chromosomal theory of inheritance. Mendel, he could not provide any physical proof. He could not provide any physical proof, neither the chemical structure of the factor. Chemical structure, what it is made up of factor. Then he is asked that where is the factor present? Physical proof means आपका ये जो factor है वो कहाँ present है? At that time, students, microscopes were not advanced. Chromosome की तो खोज ही नहीं हुई थी. Genes, no one even dreamed of. DNA, gene, chromosome, ये सब किसी को पता ही नहीं था. At that time, this genius was coming to the conclusion depending upon his observations. He was ahead of his time. We simply saw that he couldn't provide the physical proof. Neither he could say, oh, it is protein or it is carbohydrate. So no, this is also not accepted. तो ये जो प्रूफ वो नहीं दे पाया कि ये क्रोमोसोम है जिसमें मेरे ये फैक्टर्स है, ये जब पता चला, दिस इज क्रोमोसोमल theory of inheritance. That simple. Relate it to this fourth point. Now, Time passes, and in the year 1900, the three scientists, they rediscover Mendel's work. This is one thing. Come 1900, they rediscovered Mendel work and they did it independently. They were not working together. Then there was much advancement in the field of microscopy. माइक्रोस्कोपी में भी बहुत सारे एडवांसमेंट हो गए. And then the chromosomes were discovered. Chromosomes were discovered. And then VERED. And then the behavior of the chromosomes during the cell division, during meiosis was studied. Behavior of chromosomes during meiosis was studied. Then what happens? Two scientists, Sutton and Boveri, these two scientists, they found out that they were also working independently. So they found that the way the chromosomes behave, the chromosomes, they are duplicating before the cell division and then dividing again. They are behaving the same way as Mendel had said about his factors. Like the factors, they are also in pairs, and the chromosomes, they are also in pairs. During gamete formation, the number becomes half. The number in both becomes half, and after the gamete fusion, that is fertilization, the number is restored again. So what these two did, they explained the Mendel's law, re-explained the Mendel's law on the basis of observation of chromosomal behavior, chromosomal movement during meiosis, which is famously known as chromosomal theory of inheritance. Let us see the diagram. I think 5.8. मुश्किल है क्या? चलो देखते हैं. How the chromosomes they are behaving during meiosis. Okay. So this is the figure 5.8. This is the figure 5.8, which explains the behavior of chromosomes and the gamete formation during meiosis. अच्छा, एक चीज आपने कभी ध्यान दी है? When the cell division is completed, after that chromosome is single stranded. फिर when it has to divide, then in S phase उसका दूसरा वाला strand भी, then the DNA gets duplicated. तो चलो बनाते हैं. So this is G1. Fine. हम क्या क्या बनाने वाले हैं? G1, G2, Anaphase 1, Anaphase 2 and the gametes. So this is G1. We are taking two homologous pairs of chromosomes. दो ले लेते हैं. एक क्या लेंगे? दो ले लेते हैं. ठीक है? दो का minimum 2 we will take. So if this is chromosome number, suppose this is one chromosome. This is one chromosome. And this is its homologous chromosome. Same size का बना दिया, centromere same जगह बना दिया. ठीक है? Now this is another chromosome. छोटा बनाते हैं थोड़ा उससे कि समझ में आ जाए. These two are homologous and these two black one, big one are homologous. Now what? Now comes S phase. S phase में this will now form its another strand also. So now here this strand is having these two strands. Done. उसका homologous strand भी बना देते हैं. So this also done. This is its homologous strand. Fine. Now some other chromosome and its homologous strand. So this is one and this is the other. This is G2. यहाँ तक समझ गए. S में वो डबल हो गया है. DNA content has doubled. Chromosome number is same. Fine. Now in anaphase what happens? हर गमीट में जो homologous pairs है, this दो black में से एक जाएगा, दो red में से एक जाएगा. This is a very naive way of saying things. Out of the one pair of homologous chromosome, one will go into each gamete. So now during anaphase what happens? This is anaphase of meiosis 1. Anaphase meiosis 1. In this, now the mitotic spindle, what it will do? The mitotic spindle, it will, ये दोनों स्ट्रैंड समेत ये इसको अपने पोल की तरफ खींच लेगा. दिस टू विल मूव दिस साइड एंड दिस टू विल मूव दिस साइड. सो दिस वन विल मूव दिस साइड. So each one is having one of the pair of homologous chromosome. So after anaphase 1, what will happen? Two cells are formed. So we draw the two cells. ये एक सेल और ये दूसरा सेल. क्या है इसमें? One is having this one large one, this chromosome. This is also having one large one and everyone is having the other chromosome also. So this and this. Now what will happen in anaphase 2? This is anaphase 2. Anaphase 2. Anaphase 2 of meiosis. अब see here, this was single stranded. ये जो प्रोसेस है ये एनाफेज 2 में हुआ था. नाउ द स्पिंडल फाइबर व्हाट इट विल डू, इट विल ब्रेक दिस वन ऑफ द क्रोमैटिड एंड इट विल टेक इट टू दिस पोल, and then from this side also this spindle it will break the other chromatid and take it to the other pole. Same with this one. ये दो black के strands है, दोनों separate हो जाएंगे. एक इधर जाएगा, ये लाल का भी एक इधर जाएगा. Same with this one. So these two strands of chromosomes they separate. तो अब आपको मिल गए चार cells. Now you got the 4 cells. और इस 4 cells में क्या होने वाला है? There will be one single stranded red one, सबके पास एक एक, और सबके पास one strand of the black one. This way they saw that the chromosomes here they are duplicating and here the number is getting half. Here it is number 4, they are in number they are 4, here they are in number 2. In gamete the number reduces to half as Mendel had said about his factors and alleles. So this diagram easy? This is easy. Looks difficult but it is easy. You simply have to take pen in your hand and draw. 2 मिनट में बन जाएगा. Just think कि draw the chromosomes in this simple way. जैसे वो सेंट्रोमियर का पोजीशन दिखा के 2 ब्रेक करके आपके बुक में है ना, don't do it like that. उसी में आप लोग फिर धीरे धीरे फंसते जाते हो कि ये क्या है. This is the chromosome. सेंट्रोमियर का पोजीशन आपने डॉट से दिखा दिया. एक बनाया, एस में डबल, it has double. So here it is double. Anaphase में the whole chromosome without breaking the chromatids, to move this side, to move this side. अब mission formation of one chromatid. So इसके एक एक chromatid एक side में चले जाएंगे and the other chromatid moves to other pole. Then the cell will break from the middle. फिर यहाँ से break करेगा. ये यहाँ से जैसे ये यहाँ से break किया था. So ये एक cell का दो cell बन गया. From these two cells the four cells have formed. You must have observed MCQs throughout. The year of publication of Mendel's work याद रखिए. When it was rediscovered, remember that. The scientists who rediscovered Mendel's work, that also you have to remember. Then those two who gave this chromosomal theory of inheritance, in which year it was given, have I told you? This was 1902. In 1902 this was given. And then this is the behavior of the chromosomes, where they are showing this segregation. This segregation and the segregation of alleles is the same. Now one diagram 5.9 of independent assortment of chromosomes is remaining. Let us see that, and then this topic is completed. फिर से याद रखो बच्चों, क्रोमोसोमल थ्योरी ऑफ इनहेरिटेंस सुनने में बड़ा हार्ड सा लगता भले है ये सेंटेंस. इट simply gives the physical proof for the presence of the factors of Mendel. That's all. कि Mendel's के factors कहाँ हैं, इसका proof जब दिया, that is chromosomal theory of inheritance. Let us see the diagram. Diagram is that of independent assortment of chromosomes. Okay, point of caution. The first diagram we started with G1. That's why we have shown, first we have shown only single thread of chromosome. Here we are starting with anaphase. Anaphase 1. चलो anaphase 1 बनाते हैं. So this is anaphase 1 of meiosis. Of course meiosis. So this is anaphase 1 of meiosis. वैसे two pair of homologous chromosome ले लेते हैं. So but now the chromosomes will be double stranded. S phase में तो हो ही गए हैं ना double stranded. G1 फिर S, G2 फिर anaphase. Fine. So this is one chromosome. And this is its homologous chromosome. ये हो गया इसका homologous. और दूसरा क्रोमोसोम जो है और उसका होमोलॉगस, उन्हें हम दूसरे कलर से बना देते हैं. दिस इज अनदर क्रोमोसोम एंड दिस इज इट्स होमोलॉगस क्रोमोसोम. ठीक है. अब स्पिंडल फाइबर्स आप किस तरफ से निकालोगे? जो गमीट्स बनेंगे that will have one this black one also, this red one also. So आप इधर से अगर खींचते हो तो दोनों black एक साथ जाएंगे. From here if you pull it, then these two red they will move to one pole. This is not desirable na. So naturally this two, the spindle fiber, it will pull these two on this side and these two this side. Anaphase 1. अब इन होमोलॉगस क्रोमोसोम पे हम अपना जीन भी बना लेते हैं, YYRR वाला. सपोज इफ दिस वन इज हैविंग कैपिटल Y, दिस वन इज हैविंग स्मॉल y, दिस वन इज हैविंग कैपिटल R एंड दिस वन इज हैविंग स्मॉल r. चलो अब आगे बढ़ते हैं. सो नाउ दिस टू हैव gone to these poles, so the cell will break from the center. यहाँ से टूट गया, दो सेल बन गए, ऐसे ये यहाँ से टूट जाएगा ना, it will break from the center. So now in anaphase, now in anaphase 2, anaphase 2 means meiosis 2. In anaphase 2 you have two cells तो ये हो गए आपके 2 सेल. दिस इज द 1 एंड दिस इज द 1. अब इसमें क्रोमोसोम्स कैसे हैं ये तो आपको दिख ही रहा है. 1 this big black one having the capital Y and this red one having the capital R, and this chromosome, it is having homologous pair of this one which has at the same loci the small y, and here the homologous pair of this one which is having the small r. Done. Now फिर से anaphase, again they will separate. Here the complete chromosomes, the whole chromosome, से पूरा chromosome यहाँ पे move कर गया. Now 2 chromatids, this chromatid and this chromatid, they will be separated. अब बीच में से दोनों टूटेंगे. So now when the spindle fiber, they pull them, then they break open one of the strands. One strand moves this side, one strand moves this side. One strand moves this side, here also the same thing. One strand moves this side, one this side. ये भी this side, one this side. So what type of gametes you will get? चलो देख लेते हैं. 4 cells are formed. 1, 2, 3 and 4. In these 4, ये Y are these the 2 cells which are formed from this cell. Both one will have one capital Y and one capital Y and one capital R. Similar here also, one capital R. Here one capital Y. इसके दोनों, because they are formed by the separation of the chromatids of the same chromosome. Now here these two will be similar. That is, one will have this small y, this also will have small y wala, and this one will have one small r, this one will also have one small r. Is it? ये दोनों बने हैं इस सेल से और ये दोनों बने हैं इस सेल से. This is one possibility. This is possibility 1 where 2 types of gametes are formed. What 2 types? This one is yellow round, these 2 are yellow round and these 2 are green wrinkled. Just see, these 2 are yellow round, these 2 are green wrinkled. दूसरी 2 possibility कैसे हो सकती है? That is yellow wrinkled and green round. कैसे होगी? Simply this is the arrangement of the chromosomes ना on metaphase place. Arrangement में ऐसा हो ही सकता है कि ये जो small r वाला क्रोमोसोम है वो इधर खड़ा हो और the capital वाला इधर खड़ा हो. In that case what will happen? This is r, this is capital R. First we will write these two name of these 2 type of gametes ना, तो आखिर में बाकी 2 different types of gametes मिलने वाले हैं. This what we got, it is yellow round and it is, this is yellow round, this is also yellow round, this is green wrinkled. These 2 types we have obtained. Now let us see the second possibility. ठीक है? So here we have just simply exchanged the position of these 2 chromosomes. So now here it will be small r and this side it will be capital R. Same here, now in place of capital R it is small r, this is small r. This small r will be replaced by capital R and capital R. So what are these? What is the phenotype of this? This is capital Y, means yellow, and small r, that is wrinkled. So these 2 are yellow wrinkled now. And what about these 2? This is small y. Small y is green, so it is green, and capital Y, capital R in both, that is round. So you see the independent assortment of the chromosomes, forming 4 types, 1, 2, 3 and 4. 4 types of gametes. So you have studied the behavior of the chromosomes, and Sutton and Boveri, they argued that the pairing and the segregation of the chromosomes will naturally lead to the segregation of the factors also. Segregation of factors. Segregation of chromosomes will lead to segregation of factors. This also is important point. लिख ही देते हैं ऊपर. Pairing and segregation of chromosomes leads to the segregation of the factors. Chromosome separate होते हैं तो factors भी separate होते हैं. Then Sutton, he integrated the knowledge of segregation of chromosome with Mendel's laws of inheritance. Mendel's laws of inheritance. And this is called chromosomal theory of inheritance. In our Sunday session, I will again take it up. पढ़ के रखिए पहले से. Then you will find it very easy. स्टेप बाय स्टेप बच्चों, पढ़ाई कभी भी 1 महीने में, 2 महीने में, 3 महीने में नहीं होती है बच्चों. This is the means you have to start from the very beginning. So you have to start from the beginning ना. आपको आप जैसे पढ़ते चलो, पढ़ लिए, तैयार रखो in the Sunday session. Then again when it is discussed, then it will be very clear. From this Sunday, I will be also taking, one is the concept building session, another will be PYQ session. Fine. And get the link for the notes from the description box. And those of you who are new to my channel, please do subscribe, like and hit the bell icon so that you get all the notifications in time. Thank you for watching the video.